Mitosis is going to occur in four phases: prophase, metaphase, anaphase, telophase, and then we're going to move in from that point into cytokinesis. We'll go through each one of those phases individually. In prophase, chromatin is going to condense down and form a chromosome as DNA wraps around the histones. At this point, you can actually see DNA underneath the microscope. Prior to that, it is invisible to the naked eye. Well, not just the naked eye, even under a microscope, under a light compound microscope, it's impossible to see DNA because it's so loosely coiled. But once it condenses down, it's a lot easier to see and to distinguish. The nuclear membrane is also going to begin to break down at this point, and mitotic spindle fibers are going to form. They're going to play a very important role in this organizing of mitosis. The next phase is metaphase. Metaphase chromosomes are going to attach to mitotic spindles and form a line down the center of the cell. So they're going to line up right down the center of the cell. The alignment is going to ensure that each daughter cell receives one chromatid from each of the 46. This is very important. We wouldn't want one chromosome to get two number 1 chromosomes and the other cell to get, two number 2 chromosomes. Because then one cell is going to have information on hair color and how to build feet, and the other one might have 2 instructions on how to make lungs and no clue on how to make feet. It's very, very important that each cell receives a complete copy, a complete full set of the chromosomes. The next stage in mitosis is going to be anaphase. In anaphase, sister chromatids from each chromosome are going to begin to separate, and they're going to split at that centromere. That centromere, remember, is the site of attachment for 2 identical chromosomes. Once they are separate entities, the sister chromatids are considered chromosomes. Specifically, they're considered daughter chromosomes. This is not something that I'm going to quiz you over, but you will see the terminology in your textbook and in other sources if you are looking around at other tutorials. So I'm just going to help differentiate now. When attached, they are sister chromatids. Once separated, they're daughter chromosomes. The last phase is going to be telophase. In telophase, the nuclear envelope is going to form around each group of chromosomes at each pole. The mitotic spindle is going to disassemble, and we're going to see a complete separation around or 2 new nucleuses forming around the 2 groups of chromosomes.
This interphase cell is ready to enter the M phase of the cell cycle. Chromosomes have already been duplicated, and each consists of 2 identical sister chromatids. Each chromatid is one of the newly replicated DNA double helices, plus its associated proteins. In an interphase cell, there exists, just outside the nucleus, the centrosome, a cellular structure that acts as an organizing center for the assembly of microtubules. Mitosis begins with the events of prophase. In the cytoplasm, prophase is marked by the formation of the mitotic spindle, assemblies of microtubules emanating from each centrosome. The chromosomes condense into compact structures that can be seen with a light microscope. Before the cell enters metaphase, the nuclear envelope breaks down. After the nuclear envelope has disintegrated, spindle fibers from each pole of the mitotic spindle attach to one of the 2 sister chromatids of each chromosome. A tug-of-war is occurring, with spindle fibers pulling each chromosome in opposite directions. In the next stage of mitosis, metaphase, the chromosomes complete their journey to the middle of the cell. Each chromatid now faces the pole opposite that of its sister chromatid. At the start of anaphase, the link joining each pair of sister chromatids is broken. The spindle fibers shorten, pulling the chromosomes to opposite poles. This segregation of sister chromatids to opposite poles ensures that each daughter cell receives the same complement of chromosomes. In telophase, the final stage of mitosis, the mitotic spindle disintegrates, and a nuclear envelope starts to form around each set of chromosomes as they return to their original uncondensed state. Once 2 independent nuclei have formed, mitosis is complete. Meanwhile, a cleavage furrow has begun to form, and immediately after the end of mitosis, The cytoplasm of the cell divides in a process called cytokinesis, forming two daughter cells.